So, good afternoon, everybody. Thank you, Thomas, for introducing me. And thank you all for attending the seminar. I will be talking today about self-fertility in allogamous grasses, specifically about fine mapping a self-fertility gene in perennial ryegrass. This is part of my PhD research and is work in progress. So I only have some preliminary results to show you, but I will introduce you to the grasses world and the current breeding methods and the constraints on grasses, how self-incompatibility affects that, and how self-fertility also can be used to improvement of grasses and how they relate to each other. Then I will go through the materials and methods, show you some preliminary results and my expected outcomes. So perennial grasses are widely used around the globe, mainly as forage, turf, or for biofuels. As a forage, to have an idea, around 70% of the world's cow milk comes from temperate grasslands, and also a similar percentage of the beef also comes from temperate grasslands. So it's a very important component of animal production systems. Then as a turf is essential for sports fields and landscape architecture, and recently it gained attention for biofuels for its intrinsic characteristics and ecological advantages over annual crops. So currently grasses are bred as either population varieties or synthetic varieties through either phenotypic selection or genotypic selection and all the variants within those. But you end up either with population varieties or broad genetic-based synthetics, when the best cases narrow-based genetic-based synthetics, with the exception of some turf and biofuels that can also be bred as clonals. So grasses have many issues, many characteristics that affects the breeding procedure. So one of them is they are outcrossing, most of them, with a strong self-incompatibility. And other issues like long breeding cycles also affects. But what I wanted to show here in this table, I have different grass species from different countries, and you can see that genetic gains for dry matter yield, which is one of the most important traits in grasses, are pretty low if you compare with your crops or any other grain crops. You are for sure over 1% at least. And even many of them are below 0.5% annually, so it's quite low. So one of the issues, as I mentioned, is their outcrossing nature. You cannot develop inbred lines, so you cannot do, for example, hybrid breeding, which would be much more efficient. And the way self-incompatibility works in grasses, which is different from other mechanisms, from other self-incompatibility mechanisms, it's determined by two loci named S and C, and is controlled gametophytically. So the way it works, any pollen grain, the alleles at both S and C, if they are found in the stigma, that pollen grain is not able to grow and gets inhibited. So what you expect is that any pollen grain coming from the same plant will have the alleles matched, so it will not grow there. The same as very related species that share the same alleles. So this table is a summary about the species that had been characterized for self-fertility and self-incompatibility. It's just a summary. The list is much longer, but here you have different subfamilies, different tribes, and different species within them. And what you can see is that self-incompatibility coexists in across all grasses, all subfamilies and tribes. This table here is which are the species that has been characterized for self-incompatibility. So there you have self-incompatible and self-fertile species. This other column is about whether the self-incompatibility is gametophytic in control. The other one is whether the S and C system has been clearly established. And the last one is within the self-incompatible species, which ones has some self-fertile mutant reported. So what I wanted to share here is even at the genus level, you have both self-incompatible and self-fertile, the same as here for Ordium and for Lolium. Most of them are gametophytic in controlled, even though I don't recall any grass species being determined sporophytically. I guess where they didn't determine it was gametophytic, they just didn't. They just weren't able to determine it. I don't believe you have any sporophytic species there. And even this is a summary. I believe all the species that has the S and C clearly established at present here, so are actually quite few. But I would expect that all the others share the same mechanism. And about self-fertile mutants, we only know of 3 species. This is perennial ryegrass, phalaris, and rye that have self-fertile mutants reported. So how self-fertility works, in most cases it was established that it was a single gene action acting at the gametophytic level again, and only the pollen grain that carries the mutation is able to grow down through the stigma and reach the ovary. Otherwise, it's self-incompatible. So what you see is this pollen grain, which has the wild type alleles, is not able to affect pollination. So in the offspring, you can only see or expect to see either homozygote for the mutation or heterozygote for the mutation. And at the phenotypic level, what is typically seen when you cross a self-fertile inbred with a self-incompatible plant, all the F1 is self-fertile. That means they are able to produce self-seed. And once you go ahead and self that to develop the F2, also all the F2 is self-fertile. So that's why it's pretty common to see in the literature that self-fertility is dominant over self-incompatibility. However, if you look at the pollen stigma level, you see that this F1, half of the pollen grains only are compatible. The other half gets inhibited. And in the F2, you have 2 classes. One of them where all the pollen grains are able to grow, and the other half, only half of the pollen grains can grow. And this is pretty much in agreement with this model. This would be like the 100% compatible would be homozygous, and the 50% would be heterozygous. So even if it is always a single gene action, today at least 3 independent loci have been identified to confer self-fertility. And 2 of them are actually S and C, the same incompatibility loci, where mutations that can affect their functions, that can knock down the gene, and that pollen grain carrying that mutation is able to grow down through the stigma, is not recognized by the stigma. The third one is mentioned as the T locus, is independent of the other 2 and acts epistatically over S and C. If you have the wild type allele, self-incompatibility is determined by the S and C alleles. However, if you have the self-fertile mutant, it doesn't matter which S and C alleles you have. the pollen grain will be able to grow. So this is the gene we are tracking. Apparently doesn't have allelic variability. It's either that wild type or and the mutation that confers self-fertility. It's supposed to be fixed among grasses, and in perennial ryegrass it maps in linkage group 5 in a region of 14 centimorgan. And what we want to do is to narrow down this region and get closer to the gene. Sorry. So our objectives are to, well, find map the gene and find markers that are tightly linked to the gene, so marker-assisted selection can be done more efficiently. And also determine if there are other genes interacting with it to affecting the trait. So we're using an F2 population, which was derived by crossing a self-fertile plant with a self-incompatible one, and then a single F1 plant was selfed to produce the F2 population. So in this F2 population, we expect to find either plants that are homozygous for the gene or heterozygous, as I mentioned before. Any self-incompatible should appear there. For phenotyping, we're looking at the pollen stigma level to determine which is the percentage of compatible and incompatible pollen grains that a single plant produce. So for that, we're using the in vitro pollination test, which consists of dissecting unpollinated stigmas and placing them in petri dishes with growing media and pollinate them there with pollen from the same plant. So in that way, you have some control of the amount of pollen you're using and also the timing of when the pollination occurs. Then you allow them to incubate them for 2 hours at 25 degrees to give them time to the pollen grain to grow or get inhibited. And finally, you observe what happened at the microscope with fluorescent light. For genotyping, I'm using high-resolution melting system with SSR markers. If you're not familiar with HRM, after the PCR reaction, the sample is exposed to increased temperature, and at some point, the double-stranded DNA will separate because of the temperature. The software, what it does is translate that to a curve, and that melting point, or the point where the DNA separates, is dependent on the sequence of the DNA. So for every marker, different genotypes will have different curves, like the last picture here. Then for data analysis, I will have to find the marker order and marker distances, and also go through some segregation analysis, and finally find marker trait associations to locate the most probable position of the gene. So far, what we are observing is either full compatible phenotypes, and partial compatible phenotypes. The first one, you can see these lines, these bright lines, those are pollen tubes growing down through the stigma, and you cannot see any inhibited pollen there. In the other picture, you also see pollen tubes growing, but you can also see some pollen grains inhibited. They get stained and with a short and thick pollen tube, like here and here. And yeah, this is what you expect in the F2. So this graph is shows the phenotypic segregation of in that F2, and pretty much follows a bimodal distribution with one peak near 50% compatibility and the other are close to 100%. There is a little bit of an excess in the partial compatible, but it still fits the 1-to-1 segregation ratio I was expecting. So this is just some examples of the HRM output, 4 markers. Here you can see 3 different curves that correspond to the heterozygotes and the 2 homozygote classes. as well as in here. But in these two, here and here, you can only see two genotypic classes. One of the homozygous is totally missing. So it's a strong segregation distortion. I would expect that the gene is closer to these two markers than these other two. So from here, I need to finish the genotyping, complete the genotyping of linkage group 5, where the self-fertility gene is, and in other regions that could be affecting the trait. After that, I'm going to be able to analyze the data and determine if there is any interaction with other genes, and I will also have identified the flanking markers. So once I have the flanking markers, I can go ahead and genotype the entire population, which is about 1,000 plants, only with the flanking markers, find the recombinants within that region, and then phenotype and genotype with more markers, only the recombinants. And further analysis, and I expect to establish the most probable position of the gene. So summary and conclusions, perennial ryegrass has an effective self-incompatibility system that promotes outcrossing, but it also complicates any breeding methods or efforts. Self-fertility mutants are available for developing inbred lines, but we need markers that are very tightly linked to the gene so we can actually apply marker-assisted selection with efficiency. And our primary results, preliminary results, are in agreement with single gene action. And we expect with these results to contribute to the knowledge of self-fertility and self-incompatibility in grasses, and also facilitate the integration of the gene in breeding populations. And that's pretty much it. And I would like to thank present and past members of Luberstedt Lab for either helping the lab or greenhouse, and also discussions, collaborators and committee members, and my funding sources. And if you have any questions, I'll be glad to answer.
Any questions for Javier? If you don't mind repeating the question.
Okay. Yes, John.
So when you were looking at the pollen, you said that there was a fluorescence component?
Yeah. So he's asking if I'm using fluorescence for the phenotyping. So I use a dye that's aniline blue that stains the pollen tubes and the pollen grains that are inhibited. And then I observe them in the microscope with fluorescent light. That will make the dye to be bright.
So how do you just stain the ones that are inhibited?
The dye sticks to the callus, which is found, which accumulates in the inhibited ones, and also is present in the pollen tubes. That's why you see that the pollen tubes are growing. You can see the pollen tubes, those bright lines, and inhibited pollen grains are like bright dots. Yes?
Can I follow up on that question
Oh,
really quick?
yeah.
So were you just visually determining partial compatibility or 100% compatibility, or were you giving them scores?
So I'm actually counting the pollen grains.
You are,
So
okay.
yeah. So in many cases, these have been done by just saying this is full compatible or partial compatible. But if I want to determine if there are other genes, some modifiers or something, I guess I would be losing some information.
That's all done by hand, though? There's
No.
digital?
Well, this is the
processing
easiest
it?
part, actually. The
Okay.
hardest part is dissecting the stigmas and do all the lab process.
Other questions, Andy?
How repeatable the phytoplankton as to you stain the pollen? The reason I asked that question is I study heavily the soybean,
Mm-hmm.
and I stain the soybean pollen to determine whether it's sterile or fertile. So then what the time you get the pollen will affect how repeatable your phenotyping. So how about here?
So the question is about how repeatable are the phenotyping. When you take when you do the phenotyping, you do in many stigmas from the same plant, and it's pretty much very repeatable among them. About the time, I'm using 2 hours. Some researchers do 4 hours after pollination. But this happens really fast in grasses. So I guess there's no need to wait for too long. And also, if you wait too long, all the pollen and everything starts to degrade. So that may confuse your results.
And for the soybean world, they say when we decide whether it's sterile or fertile, we need to study very carefully, because even though you have 1% of the pollen is available and may provide enough pollen, so we give a lot of pollen. So here we will be determining like how many percent of the pollen is available, what is threshold you call the seraphim compatibility, or what is the threshold you use, so it's 100% or what?
Oh, I don't know if I get your question, but either one of even if it's partial compatible, it will be able to produce self-seed. I mean, you only need one pollen grain to grow down.
So let's say you check all the pollen at the 100%, this stay, and then you call it incompatible.
No, I actually mentioned the percentage of compatibility. That's yeah.
You mentioned that this region that you're trying to define marking is 14 centimeters, right? So my question is, how many markers are you using? And the other question is, do you have an idea of how many base pairs are in those 14 centimeters?
So the question is how many markers I'm using in the 14 centimorgan region and how many base pairs, if I know. So I don't know how many base pairs there are. It's a huge region. So and I'm using about 6 markers in linkage group 5 that are surrounding that region. And so these 4 markers here and the other ones that are outside. And then I'm using more markers in other regions, just to track if there are any interaction. So here I only need to establish which are the flanking markers. That's why I'm using only a few. But then after I have the flanking markers and find the recombinants, I will add more markers in the middle. How many, I don't know yet, but it will be a few, not too many. Yes.
Javier, you said you also have some markers in other regions in order to track in any direction. And how you select that marker? Is random?
So there's a previous study on this that showed some segregation distortion in linkage group 3. So I'm using the same markers as in that study. And also markers for S and C, which are in linkage group 1 and 2, to see if there is an interaction with them.
Can I ask you a final question?
Yeah.
How did you decide that 1,000 individuals will be enough?
So the question is how I decide about the total population size. Well, it's just a number. I don't know. I can go with more individuals if I need, or less if we have too many recombinants in the middle. It's just a starting number.
Javier, do you have a working hypothesis on the mechanism behind this compatibility at all?
You mean the biochemical stuff behind it?
What causes the pollen to arrest?
Well, In grasses, we're a little bit behind other species, so we don't know which are the gene products or pathways or anything. There have been some efforts on fine mapping and trying to, well, also determine the gene products. And there are some advances, but no system has been unraveled. So I can tell you.
I'm sorry. Is the S and Z is also in linkage group 5?
S is in linkage group 1 and C is in linkage group 2, sorry.
So then the whole, you said, if I understand correctly, the presentation, we have 3, you call it single gene, but it's like 3 loci.
Yeah, I mean,
But they are in different chromosomes.
yeah. So maybe I wasn't clear. Any of those genes can produce self-fertility, acting independently. You may have mutations at both of them or the 3 of them at the same time, but it's. I mean, mutations can affect self-fertility at either the 3 loci, but just a single of them, just one of them, is able to produce self-fertility.
I thought you should say a little bit about what the challenges are to move from population to hybrid breeding. So even if you master this self-fertility and have closely linked markers, what still are challenges?
So the question is about what are the challenges to move through hybrid breeding. So I guess the first step is being able to develop inbred lines. You may have some issues there with inbreeding depression, but I may anticipate that would be not something that breeders cannot deal with. Then another issue is at the end, once you have the inbred lines, is you need to make crosses without selfings. Now these plants are self-fertile, so you will always have some contamination. So ideally, you may want to recover self-incompatibility at the end. So you can then put together a bunch of seeds from different lines, plant them together, and then harvest everything. Everything will be a hybrid. But the problem is that you get stuck with this. It's not easy to recover self-incompatibility, at least at a commercial scale. You know, you can have a self-incompatible inbred, but at some point you will need to multiply that to a large scale, and now the inbred is self-incompatible, so you cannot produce seed. So another way would be to introduce male sterility, which is the way many other crops have solved this issue.
Sounds like there'll be a couple more steps after you're done.
Yeah.
How many students are going to work on it?
Yeah, exactly.
I don't know.
As the funding goes, more projects get added. Well, Javier, thank you for that. Let's give Javier a round of applause. Any bad words? I'm going to quick introduce Kyle because his mentor, Dr. Danny Singh, cannot be here today. So we can beat up on him as much as we want to. Like to. Kyle joined the department how many years ago?
In 2014.
In 2014, he got his bachelor's degree at OSU and is now here working with Danny Singh on his MS degree, which he will talk about today, but he's also doing additional research looking at identifying, using high throughput phenotyping, physiogenetic factors for yield in soybean at different row spacings, which he presented for his poster, but now he's going to today talk about his research that he did for his master's degree. So let's give Kyle our attention.
All right, thanks. Can everybody hear me back there? Great.
Wait, I didn't know. Oh, it is. Is it good? Yeah?
Good.
Okay.
All right. Yeah. So again, my name is Kyle Parmley, and I'm going to talk to you about my master's project, a QTL mapping of seed oil and protein content in a really diverse biperennial soybean population. A little bit what I'm going to talk about. I'm going to talk about soybean production and the seed content and its effect on how production is changing and how production is changing the composition of the soybean historically and currently and into the future. Talk a bit about some of the products that we derive from soybeans, what are their market importance, talk about the historical, and then get a little bit into my research, kind of where I'm at. This is my preliminary results, so I'm still continuing some results. So to just introduce the really big importance of soybean, soybean accounts for the majority of the world's oilseed crop. In fact, over 50%, and the next biggest is canola with a marginal 14%. And this is really due to just the vast production and economics of soybeans. Soybeans has a really good amino acid profile, and it really makes a high quality feed product, as well as a really cheap oil product used in human and industrial uses. So a little bit about kind of where the product comes from in the seed. The seed, this graph here on the top kind of breaks down into the seed components, with the top bar here being the entire seed, about 40% protein, 21% oil. That's a typical 2-to-1 ratio that you'll see in most soybeans, with the majority of some carbohydrates. What I would like to point out is the majority of what we extract, or where the protein oil comes from, is in the cotyledons. And here, obviously, this image shows that here. The cotyledon makes up most of the mass of the soybean. And then there's other components have different compositions. I would like to point out this past year, the most recent data that's out there, in 2014, protein was actually down that year. And it's 34% protein and a little above 18% oil. So to talk about the flow of soybean products, how kind of the rough process, how it works. Soybeans that would come in from a farmer's field go through a process what's called crush. You would hear this number being thrown around on the market floor, and from that, and actually soybeans are actually physically crushed in this process. It's literally what it sounds. And from that, we have two main products, soybean oil and soybean meal. Soybean meal has to be dried and then is a really high quality feed product. That soybean oil can then go to food use to fry some of your foods, or even into biofuels, and the list of uses is growing for that product. Here on the right, the two pie graphs that I have here, this breaks down the usage of those two products. You see oil, the majority is used domestically, and then protein, there's the majority is still used domestically, but there's still this portion that is exported. And I'll kind of talk a little bit about the importance of that, and that's susceptible to this world market, the global marketplace that's out there. Here's a figure showing the U.S. soybean utilization, how we market our product. On the y-axis, there's in millions of bushel and in years. And in the blue color, you have the amount of soybeans that are dedicated to crush. And you can see that takes up the majority of, or not the majority, a good amount of the soybean production. And then followed in orange by the amount of soybeans that are exported. The U.S. exports a little over 30% of their crop, so that's a very significant portion. And then the other, the yellow color and the orange are what's left over, and then some seed and other uses for soybean. Here in my next graph, kind of getting into the importance of seed quality and its effect on the world marketplace. And again, we have, in this case, we have million metric tons a year. And draw your attention to the blue bars. That's actually the U.S. soybean, the amount of soybeans we're exporting. So if you'll notice, it's kind of an increasing trend. That's all great. But when you look at this on actually a world scale, and I'll draw your attention now to the purple line, the U.S. is actually seeing a decrease in the share of total production, because you have these other places around the world that are improving production, but yet the U.S. is kind of plateauing out, and I'll show that in a few slides. And it's kind of remaining the same. And there's some small increase in soybean meal, but nothing too significant. Here's a graph kind of showing the real competitiveness of the different countries throughout the world, and kind of some challenges that the U.S. is facing to market their product to the world. And again, a total amount of seed in years, and in the blue color you have the United States. And this is being projected from 2010 on, and I believe that goes to 2019. And if you'll notice, the U.S. it's pretty flat, kind of a plateau effect. And while in the orange is Brazil and the light blue is Argentina, you see these South American countries really starting to increase the amount of soybeans that they're putting on the world marketplace. And it is this figure here that I'm really trying to get across, that as U.S. producers, we need to think about our soybean quality to really put a value-added product out there in the world marketplace. So as producers have changed the areas of production to where we produce soybeans, the top image here is taken of soybean acreage in 1963 through 1967. Nothing that you probably wouldn't expect. There's majority of acreage is in the Midwest, pretty typical. But as we have more current times, you see this huge amount of production that's occurring in these northern areas. And we know that these more northern areas these different regions have different capacity to produce different amounts of protein and oil. And again, I'll show this in the next slide. So just as different regions have different temperatures, different growing environments, we know seed content has different responses to temperature. Here in these two graphs here, I'm showing oil on the y-axis and protein on the y-axis, and mean temperature. And you can see this this increasing trend here in oil up to about 22 degrees Celsius, and then it plateaus off, and you see this curved response for protein. But when you put those two together, and this is protein and oil and mean temperature, it's a linear response. So if you think about where we're producing soybeans now into more in Canada and North Dakota, those are cooler environments. So we're going to be more down into this portion. Where if you go to Mississippi, a southern environment, you're going to be really pushing this because your temperatures are going to be quite higher. I want to spend some time on this graph. This graph is kind of, there's a lot going on here, and it's simply oil and protein. But I want to draw your attention is to the kind of the middle shaded area. And what that is that is the combination of oil and protein that produce acceptable protein meal that we can put on the marketplace. So anything to the left is lower, and it would be at a discounted value, and anything to the right is just icing on the cake. And you can see that this Minnesota and Mississippi, but Minnesota is becoming critically close to that cutoff, if you will. And it is actually, this is kind of a good example of what is happening to soybean production, because as we continue to move production northward, we're going to be approaching this kind of this boundary effect, where in these more southern environments, it's not an issue. And some other factors that we know that are affecting this is seed yield. We know seed yield is negatively correlated to protein and is slightly positive to oil. But these regions in combination with this association with yield is really affecting the quality of soybeans that are being put into the world marketplace. And kind of a summary of what I've been telling you is currently producers are compensated just on a by weight basis. If you were to take your semi to an elevator, they're going to weigh it. You're getting paid for that weight. And to provide an incentive to producers, we really need to start thinking about the protein, the composition, amino acids. There's a whole list of other traits we could add that may help incentivize producers to produce a higher quality product. So when we're shipping these giant loads of soybeans, it'll be a high quality product. I added this, ran across this actually today, and there's a survey that was conducted around the Midwest, and this is done by the U.S. Soybean Checkoff. It suggested that 69% of the producers believe that value-added soybean meal, so high amino acid, high protein, it was just a general question, that they should be on top priority for the checkoff. So this is something that farmers are starting to recognize, something that needs to be corrected. And then finally, where I'm going is that hopefully this would increase exporting demand because we have a value-added product. I want to touch a little bit about the genetic diversity. We know that there's been a severe genetic bottleneck in elite soybean germplasm, and actually 80 of the 45,000 land races account for almost all of the parentage of North American cultivars. And this is a quite amazing statistic. 17 of those 80 account for 86% of the parentage. And and the rest have less than 1%. So there's really this really urgent need to incorporate new diversity, not only for seed composition, but for other traits. This is some example of some unique diversity that's out there. And in fact, some of these look more like weeds than they do soybeans and act more like weeds than they do soybeans. So this gets me to the objective of my project. Just as the title suggested, was a QTL mapping in a really diverse real population for just protein and oil content. And this was done across 2 locations, Champaign, Illinois, and here at Ames, and across 3 years as well. So this was the breeding structure of my population. I had 2 PIs. They come from diverse areas around the world, one from China, the other from Russia. Those PIs were identified as potential sources of high oil or protein in a test of 57 PIs in 2006 and 2007 at Champaign, Illinois. And this cross was done in 2008 and 2009 in the greenhouse in the winter, and it was advanced from the F2 to the F4 in a single seed descent. In the F4-5 generation, it was initial test was done for oil and protein to see if there was transgressive segregation. There was, and so they advanced that population, and from out there was 118 reels that were created. This is a picture of my reel population taken from this past year, and you can really pick out the diversity. I'm standing kind of on the border of the field. You can notice the elite line, the really high chlorophyll content, and if you'll notice, the leaves are very skinny, very long, and so they really have a wild characteristic to them. And you can even pick out some elite checks that I have in there. This is a layout, the table that I have here, a layout of the experiment, a location and the design. Again, it was Champaign, Illinois, and Ames, and this was repeated 2013 through this past year. All done in RCB except for the first year where they had 1 rep, and done in 1 rows up till 2015. We decided 2 rows, 2 short rows, just to facilitate harvesting. And then, but I will not be presenting data. I just received data from this 2015, so I won't be presenting on it today. These are again seeded at 140,000 seeds per acre, and then I collected height, lodging, maturity throughout at R8, and but I will not be presenting QTL for those traits. It's an example of some of the issues that we had during the growing season. This picture is severe lodging. It was occurred in this population. And this was about the first week of August that occurred. So these plots were harvested. The entire plot was collected for NIR using this the FOSS Infratech 1229 just across the street here, and just protein and oil composition as well as fiber was collected, and that was adjusted to 13% moisture. I'm currently using JUMP version 12 right now from all my analysis. So some of the data that I'm going to show you today, I collected a single location ANOVA, a combined ANOVA with all locations, excluding that 2015 Illinois location. I conducted a linear regression to determine the relationship between protein and oil, and then LS means were done for the QTL mapping. The soybeans, the reels were genotyped in the F4 generation, excuse me, the F5 generation here at Iowa, and using the SNP 6K chip. And of those approximately 5,400 markers, I found 38.6% were polymorphic. The other markers were removed. Of those 38.6%, 5.3% were in distortion, another 2%. The parents had a heterozygous at that loci, and so those were removed as well. I used ICIM QTL mapping to construct my linkage maps as well as do the QTL mapping. It's a free software, and I was able to learn it and utilize it. In my linkage mapping construction, I used 843 markers, and so these are the parameters I used to construct those linkage maps. I used a LOD score 3.5, and in ordering I used a traveling salesperson algorithm. It's called NN2opt. And then for the QTL mapping, I used their, they call it the inclusive composite interval mapping, and a LOD threshold of 3. Here I'm showing the distribution of my population. This is the combined data, excluding that last location. And you can see I have really good transgressive segregation. This is a protein here, and both parents have very similar protein composition. I had this table showing their composition here. And on the right is oil. Again, very close. Out of this, I'd really like to draw your attention to there's some really extreme values. When I first seen these, I really had to double check to make sure those were real values. They come out as outliers, and those are real values. So we really have some extremes in this population. Here I'm showing the averages for each location. There was a location effect, and 2014 Illinois was the highest protein and oil combination. And this table here, there was significant differences between all genotypes for protein and oil. Nothing that we already don't know. We know protein and oil are inversely related, just depending on the slope for some populations. I've seen a slope of 0.94. That was been reported in literature, so nothing out of the norm. What I would, again, draw your attention to these really two extreme points. Those were those same two that you've seen on the distribution that were really far apart. And these two genotypes could possibly be used to really drive directional selection. Here we have one that's almost 40% protein, the other is really low protein and high oil. These two genotypes really spell out kind of good examples of the inverse relationship between protein and oil. And here's I really have a good group of high protein, high oil. And these could be used as potential parents in a breeding program that could introduce QTL for both protein and oil potentially. So here are just some of my preliminary QTL mapping that I've done. In the black text here, these are QTL that I detected one time, and they weren't repeated. And in the blue here is QTL that I've repeated across years and location. And the first QTL I'm showing is on chromosome 8, and it has a negative additive effect. So that would be and that additive effect is the difference between parent 1 and parent 2. It's not necessarily a negative effect. It's just a difference between the two parental alleles. Again, I have another QTL I'm reporting on chromosome 9 that, again, across locations, across years, and have a positive effect. And lastly, I have last QTL I'm reporting is on chromosome 11. And there's kind of a unique response that's going on here. This is just in Iowa. So there's obviously a location interaction that's occurring with this QTL. And for this year, 2014, there's a negative effect here, but then a positive effect for those two locations. So that's an interesting QTL I'll be looking into. So just wrapping up, we know that seed quality is affected by environment where it's grown. As you move north, you should expect lower protein, lower oil, and then as to really try to improve demand, we need to try to increase this composition to create a value-added product. And then we know that we need new genetic diversity, but not all diversity is good diversity. And then lastly, I did identify QTL. So some future work that I'm going to be conducting pretty shortly, I'll be doing the QTL mapping for the last location I just received the data for, and then the co-localization of other QTL that have been reported in soy base. So hopefully we find these QTL that have been detected and possibly some novel QTL. And then I'll be doing an epistatic QTL mapping and multi-environment QTL mapping. And with that, I would like to thank the people that have funded me, R.F. Baker Plant Center, Monsanto, Iowa Soybean Association, and the Seeing Soybean Breeding Group. Many of you are here. And with that, thank you.
Wonderful job, Kyle. Any questions for Kyle?
See.
Is there any yield to oil and protein ratio? So if you see yield increases, would you see the percentage change for oil and protein?
Sure. So if as yield changes, what is the response in protein and oil? So I guess it's as we improve yield, I guess like a hypothetical example, if we had a line that yielded 2 times that but had half the protein, you would have the same amount of protein across that acreage. Is that kind of what you're asking? I mean, so cumulative protein. The issue is when you get to such low protein values, you really start decreasing those amino acid composition. So you may still produce the same amount of protein across the area, but the quality of that product is diminished.
So is there association between Q-trait? As you see yield increase, the protein percentage will actually decrease?
Oh, I see what you're saying. So yeah, we know protein and yield are inversely related, but yield and oil there's a linear increase in protein or in oil and yield. Yeah.
When we make a matching population, we usually choose 2 extremes, like very resistant, very susceptible. We will choose those 2 PIs. Their oil and protein are very similar. What is the theory behind it?
So I guess the question is, why did we use the 2 parents? The 2 parents are very similar. I would agree with that. But when you look at these 2 parents, when you compare them to the elite germplasm that's out there now, they really stand apart. I can't speak on why our group did not make those crosses. Randy Nelson at the USDA did. But it's interesting we are getting QTL, and that may speak to the fact that they come from different areas in the world.
Yeah, China germplasm gene bank, we do similar work, but we will choose the highest protein or lowest protein, make the population
Sure.
soil similar.
Yeah.
I never see the population, they have very, very similar both, and you got huge segregation.
Sure.
And surprising to me.
Yeah, no, there's many papers I've reviewed that have actually find those 2 extremes, but when they create the population, they can't find any transgressive segregation. So it's interesting that we're getting that in this.
The other, can you explain a little bit more your cross year, across the location, QTR?
Yeah, sure.
With the single year, single location, the table, you have 3 tables?
Yeah, so I'm not changing, I'm kind of adding as I was going through that presentation. So the QTL that are in the black text are just QTL that were detected once, so not across locations or years. Basically, any QTL that are in colored, so the blue are QTL that was detected 2 different years at 2 different locations, and they're both.
If I read it correctly, say from Illinois, you got QTR from chromosome 1313. From Iowa, Booker, you got 76, and from Iowa, you got 8 and 9. But when you do a cross here cross location, you got chromosome 11. Do I read your table correctly? Yeah, this is, to me, the inconsistent results. Because say if I have a QTR unidentified in that location, maybe I have 3 QTRs unidentified in that specific location, and then another 3 QTR unidentified another location, another year. Then you put a year, all the year location together, you will find overlap QTR. But what you find here, when you do it from different location, different year, you find QTR from different chromosome. When you put it together, you find another chromosome, which is 11. It doesn't make sense to me.
I guess I don't, we may have to talk later. I don't understand your question. I don't. Tara?
So in this graph, are these all results from single location?
Yes, yes. So these, and I didn't say that each location was analyzed separately. So this isn't a combined analysis.
Did you run that?
No, I haven't ran that yet. I didn't want to run that until I had the last years of data, which I just got, the last location. So maybe that is kind of where you were going. Sorry, yeah. Yeah.
I was just curious about the agronomic characteristics of the carrots for the massive population.
Mm-hmm.
How do they compare with
So, yeah, my parents look very similar. They have very long leaf. In that picture I showed where they're lodging, I don't know, maybe you're pretty far back there. They have, I wouldn't call it terrible, but not good agronomic characteristics. They actually seem to yield decent, so there's maybe some attributes there. The seed weight is probably below average compared to the elite genotypes. Yeah, not great. So.
Are you planning to do, so you analyzed all these locations, you analyzed these each by location?
That's correct, yeah.
So are you going to do like a meta-analysis to combine everything, or?
Yes, so that's the multi-environment QTL that I mentioned in my future work slide. So right now, and I did this all because I didn't have that last location of data. And so I analyzed each location separately, and I'll put those together. But I did have a location effect, so that'll be, I'm not sure how that'll affect the QTL mapping.
So you use the interval mapping, right?
Yes.
Why your result is only pinpoint like specific location?
I took the left and right marker out, the flanking markers out. It was just taking up a bunch of space. Yeah, I guess that is kind of confusing.
Are you afraid of the Bemis effect?
I think in this population, yes. Since we're dealing with parents that are both high protein and high oil. Yeah, and since they're coming from a diverse line.
Also regarding the population size.
Yeah, that was an issue. In fact, initially in 2014, I had 8 populations. And I chose this one because it had the largest population size. And so I didn't have a whole lot to work from. The detection power is really low. But. Okay, sure. So this is it's done with near infrared, and this machine here on the bottom. I don't know a whole lot about the intricate details. In fact, that's going to be a good thing to know what wavelengths it used. But this is a NIR machine. It's a whole bean analysis. You don't have to ground. You can grind them. It's literally using certain wavelengths that are calibrated for samples of different protein and oil composition that are, I guess, off reflectance.
People use, is it primarily NIR that people are using for?
Yes. Yeah. So.
Not NMR for oil content?
No.
Okay.
No. NIR is the predominant. You see a lot of different names, but it's all NIR.
It's all.
Yeah. No, you're fine.
You are
No. 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 And that's kind of it's a good question, because we know in a lot of livestock, like pigs and chickens are two that are critical for, there's 5 amino acids that are really critical for their production. And so that's a big area of study that kind of has been untouched. And no, just for this project, it was just content. And I feel like we got to get that conquered before we go on to really compositional.
Are you looking at seed size in the soybeans to see if there's any difference between large seeded and small seeded, and if that there's an effect between like
Sure.
zeros to later maturity?
Yeah, so as far as I really am not. I have seed weight data, so I could look at that. That was, so that's something I didn't plan on looking, but that would make sense. Have a larger cotyledon, you could put more in that.
Yeah, that is a covariate.
Mm-hmm. Yeah.
Any more questions?
Yeah. What is RM of the parents?
They're both group 1. They're late group 1.
It's like all of them, like how far away, 5 days away, or
They're probably less than that. I would call them plus and minus 2. I didn't show, but I actually had a picture of them very, very close to maturity, and they're almost 2 photocopies of each other. Yeah. For being from diverse sources, they're very similar in their phenotype and agronomic characteristics.
Alrighty, well, let's thank Kyle one last time. And if the students in 600A will stick around for a little bit, and anybody who has additional comments or suggestions, that'd be great too, or just pass in your comment sheets. And thank you for coming. Good job.